Good day to everyone joining us, and welcome to today's X-Talks webinar on using amorphous spray dry dispersions to develop oral solid dosage forms. My name is Zaita, and I'll be your X-Talks host for today. Today's presentation will run for approximately 60 minutes, followed by a Q&A session with the speakers. At this time, all participants are in a listen-only mode. This event will be recorded and made available for future download. The presentation slides will advance automatically. This webinar is designed to be interactive, so please feel free to send in text questions and comments throughout the presentation using the questions function located in the question panel on the right-hand side of your screen. If you need any further assistance, please contact me at any time during the presentation by sending a message in the same question panel. We would now like to thank Ben Research, who has helped develop the content and presentation for today's meeting. For more than 35 years, Ben Research has worked with clients to solve their most difficult scientific and technical problems. The company's success is based on a strong understanding of science and engineering fundamentals. Ben Research has numerous capabilities to support the advancement of promising drug candidates from conception through commercialization. This includes formulation and dosage form support, process development and optimization, and cGMP manufacturing. Ben Research is a leader in a novel drug delivery technologies such as spray-dry dispersions, hot melt extrusions, and controlled release, and has expertise in inhalation and biotherapeutics development. Ben Research employs nearly 300 people and has 6 state-of-the-art facilities located in Bend, Oregon. For more information, please visit www.benresearch.com. I would now like to welcome today's speakers. Chris Craig is a research fellow at Ben Research, Inc. His expertise spans physical chemistry, process, and pharmacokinetic modeling, engineering design, formulation, and process development and scale-up. His knowledge in the areas of spray-dried dispersion fundamentals and dosage form supports a variety of client projects. He has developed immediate and modified release dosage forms involving dry granulation, direct compression, particle size reduction, cosmetic and functional pan coating, and laser drilling. He has also supported the installation, qualification, and performance qualification IQ and OQ on the site in cGMP manufacturing sites. Mr. Craig holds a BS in chemical engineering from University of Rochester. Also joining us today is Randy Wald. Randy Wald is a senior research fellow at Ben Research. Randy Wald is a director of oral drug delivery technologies at Ben Research. He oversees formulation and process development for laboratory, pilot, and commercial scales of immediate release, modified release, and enhanced solubility oral solid dosage forms, including those incorporating Ben Research spray-dry dispersion intermediates. In his capacity, he also supervises the technical transfer to cGMP manufacturing sites such as Ben Research manufacturing and client and partner CMO, CDMO sites. Unit operations commonly used include fluid bed processing, wet and dry granulation for immediate and controlled release tablets, wet milling, and dry particle size reduction techniques, twin screw melt extrusions, encapsulation, single and multi-layer tableting, and both aqueous and solvent film coating. Mr. Wald holds a BS degree in pharmacy from the University of Minnesota. He holds 2 patents, has written 5 articles, and has given more than 20 external presentations. Participants can access more information about our speaker and sponsor at the event's main page on www.xtalks.com. And now, without further ado, I would like to hand the mic over to Chris Craig. Chris, you may begin when ready.
Thank you, and thank you everyone for joining this morning to discuss using spray-dried amorphous dispersions to develop oral solid dosage forms. Ben Research, as mentioned, is an employee-owned company of nearly 300 employees located in Central Oregon. It consists of 6 vertically integrated sites, allowing us to go from research analytical all the way through to cGMP manufacture to help our clients solve their problems with drug delivery. Jumping right in, so one question is why amorphous solid dispersions? There certainly are a lot of technologies available to help solve enhancing API solubility and delivery for low soluble compounds. We've found that spray-dried dispersions offer a variety of advantages, and we have listed here some of those advantages. We find that they're broadly applicable to a diverse chemical compound space, and often very simple with the binary systems, and they're also very well tolerated. They are amenable to solid oral delivery. So the dispersions here is shown on the slide here. Have been used, and as a mature technology, it's been used for a variety of compounds, and it's been helped to advance through feasibility assessment all the way through to phase 3 supplies there. The graphs show just basically representative data here that the SDDs can provide an enhancement over crystalline cases, and in this phase 3 case, provided enhancement over a soft gel. The graph on the right is just to illustrate that the chemical space, it's a rather diverse chemical space that can be applied to with amorphous solid dispersions. Amorphous solid dispersions are not therapeutic specific. They are applicable to a broad chemical space. So the process for developing a spray-dried dispersion involves taking your API and dissolving it up in an appropriate solvent along with your selected polymer. What that does is allows you to break up the crystalline lattice forces there by dissolving it. And then what's happened is you spray dry the material, and what you're doing is flash removing the solvent, and allows you to set the amorphous state with the dispersion. As shown on the right there, you get from DSC, you can get analysis there, and you'll get a single glass transition temperature indicative of a homogeneous amorphous material. The SDDs provide bioavailability enhancement through disintegration rapidly, rapid dissolution in the intestines, and an increased solubility. If the dispersion is designed properly, it'll also allow for sustainment of that super amorphous solubility throughout the absorption window. What we have here is a list of common dispersion polymers that are often used for spray-dried dispersions. There are a variety of polymers available, and the polymers that you do select are dependent on the API properties and what you're trying to get there. There are both ionizable polymers and neutral polymers. A particular polymer that we've found to be extremely useful is HPMCAS, which I'll discuss here. HPMCAS has a variety of key attributes that make it a very nice polymer to use for amorphous spray-dried dispersions. One of the key things is that it's amenable to oral delivery platforms. It also, because of the chemistry of the polymer, allows functional enhancement of the sustainment of the amorphous free drug. The high glass transition allows for stability of the amorphous state in both in the solid state. The limited water uptake, as you can see on the DVS curves here in the lower left, there's a limited amount of water that it takes up relative to, say, PVP, for example. That limited water uptake correlates, as you can see on the right here, to a higher glass transition temperature when exposed to water. And the glass transition temperature is a very good first tier indicator for solid state stability. So the HPMCAS, as shown in this example here, would allow for better physical stability at accelerated conditions listed there, like 4075, for example. Yeah, and so the another key feature to the HPMCAS is that when it actually gets wet, the dispersion there can speciate into a high energy form of the amorphous material there, which is the key to providing the high free drug, which is, of course, the requirement and what we're looking after here in order to get good absorption in the intestinal tract. So what's a key feature here is that the SDD rapidly form and disintegrates to form colloids, polymer drug colloids, and then also to source the free drug. And at this point, what we've done is we've generated a powder of the spray-dried material. And when we have this, it's a high surface area material. And then when we generate a dosage form, what we want to do is be able to preserve all the features that we have put into the material through the spray drying process. And so that acts as our basis for what we're trying to do when we try and make a solid oral delivery for the dispersion. Some common dosing options for SDDs include suspensions, immediate release tablets, IR capsules, and of course spray-dried dispersions are also amenable for controlled release, which can be done through osmotic tablets, here listed as swellable core technology of push-pull systems, or also hydrophobic, as you fill it, matrix tablets. Some important things to note for the suspensions. The suspensions are very good for initial early toxicology studies and for in vivo there. They're not often useful for progressing towards commercialization. That's where we move obviously into immediate release dosage forms, which will be the bulk of the talk today. Also capable is dispersions are amenable to IR capsules. An important thing to note with an IR capsule is that it does require formulation in order to typically work there. You can run into problems if you take unformulated SDD and just place it into a capsule. We have here on the next slide is an example of taking an HPMCAS SDD and just placing it into a capsule. What can happen is as the SDD placed into the capsule, the water as it dissolves the capsule can actually cause the polymer to stay closely associated with itself there, and as the water comes in, it can potentially gel the material. And so what you end up with is a a large mass that has a low surface area, and it becomes difficult for the rapid dissolution. What you can see in the graph in the lower left is a two-stage dissolution test where the material is placed into gastric media at pH 1.2 and then is there for a half hour and then transferred over. The solid blue line is the SDD controlled as made as a suspension, whereas the dashed line is a capsule that was just placed into the USP dissolution vessel. And you can see a much slower dissolution performance here. And this slower dissolution performance would transfer over to potentially not gaining the advantage in vivo. If you take and you reformulate this dispersion in with some disintegrants and some additional fillers, what that allows you to do is just to disperse the and granulate the material. Allows you to disperse the powder better when it comes into contact with the water. Then that's allowing the dispersion to go back to its native particle sizes and allow it to dissolve and take advantage of the small particle size in the rapid dissolution. We have found that although capsules do work well if they're formulated, that by the time that we've gone through the work of formulating the capsule, that we might as well almost just formulate the tablet, because the tablet is almost is normally the ultimate goal for most of our dosage form work is to deliver a tablet for the further downstream. So our goal in developing a tablet is to, as rapidly as possible, get the tablet to disintegrate, macro disintegration, such that it reforms the granules and then back to the native SDD particles as rapidly as possible, such that these formulations can then be within the absorption window, and we can take advantage of the high energy nature and get full drug absorption. In here we have an example of a test that we like to use for determining the performance of tablets within solid spray-dried dispersions. What we do is like to take advantage of a two-stage test where you place the material in a gastric media and then transfer that into an intestinal media. And it's important to note here that what we're doing is we're testing these in a non-sink environment. And during development, we like to use a non-sink test such that it allows the dispersion to be tested under a case in which it still needs to perform, and you can allows you to see if during the tableting process you've done anything that might adversely affect the performance of the SDD. And of course, as you move later on into downstream processing, we do normally develop a sink test. But for the initial work, what we like to do is to have a non-sink test. So that way we can determine if we are getting the rapid disintegration of the tablet followed by the dissolution of the SDD. On this slide here, what we're allowing us is to start looking at some of the practical aspects of taking a dispersion and putting it into a dosage form here. On the graphs, the axis here is the dispersion SDD loading in the tablet, and then on the y-axis, and then on the x-axis is our dose, desired dose. The graph on the left is calculated for a 25% active SDD. The ISO lines represent here the resultant tablet weight for a given dose and loading in the tablet for the 25%. The graph on the right is for the 50%, is corollary for the 50% active SDD. Here what we're able to show is that in lower loading in the dispersion generally allows for a higher drug loading in the tablet, and that's because the dispersion takes on more of the polymer nature than the API nature there, and it actually requires a few less excipients to get the materials to break up appropriately. As you move to a higher loading, the API itself can start to play a larger role in the tablet disintegration. Certainly high loading tablets and even higher loading dispersions are capable, but listed here are sort of the generally very easy to formulate ranges here, and the possible to formulate are done frequently, but you start to move to a little bit more to a case by case when you have depending on the nature of the API. At this point, I'd like to transition the presentation over to Randy.
Thanks, Chris. So we're going to transition over a little bit from talking a little about the SDD technology in of itself, and Chris mentioned how that starts marrying up with the tablet. In the case that he used here, this 100 to 200 milligram tablet dose is probably the most common and that has to do with primarily because of the fact that most of the amorphous solid dispersions that have the optimal physical stability as well as performance in vivo or in vitro, I guess, initially, do fall into this 25 to 50% active range. So the tablet sizes here, typically are in the 400 to 800 milligram size. So I'm going to talk a little bit more about the process here and then how we go about developing formulations from inception up through at least through clinical manufacturing at pilot scale. So one of the key things to note here is really our objective is sort of to do no harm. Chris was mentioning that we've put a lot of effort into forming a stable, well-performing drug product intermediate. And our goal then is to deliver that effectively as if it were a suspension. That's sort of our ultimate goal in almost all the cases. There are some exceptions where we'll get too fast of an onset or something where we want to do a little bit slower. But generally speaking, it's try to get it the disintegration and dissolution as fast as possible to make sure that we don't miss any absorption windows where the bile salts and the highest absorption occurs. So we've been doing this a long time, and it really has worked in to be somewhat of a platform nature. And I'll talk about some of the properties why that is. But so our philosophy is we do a development and select our formulation at benchtop, and then we do once the composition and the process are identified, we do kind of a confirmation on the process equipment that then ultimately would initially go into clinical manufacturing. So what I've shown here is the kind of the pilot scale, lab pilot scale process trains that are used almost exclusively for the spray-dried dispersion technology. We do find the dry granulation is most amenable. We try to keep it away from aqueous environment, although wet granulation is possible for many of the SDDs that we formulate. The a dry process is more amenable. Additionally, both of the dry granulation and the direct compression processes shown here are continuous unit operations that are very good, are very useful and easily scalable. So if you notice, this is really not any different than if this were an immediate release non-solid dispersion API. Bin blenders, twin shell blenders typically used. Roller compactors or direct compression, and then encapsulation, either standard or bilayer tablet presses or standard tamping or dosator encapsulators. One thing to note is aqueous film coating is certainly, we do it very routinely. dispersions are stable enough to withstand the aqueous coating environment. So I've listed on this slide here, I'm probably not going to read all of these, but some of the key positive attributes. It turns out that, at least for these enteric in particular, for an HPMCAS SDD, there's some attributes that are really nicely amenable to formulating into a tablet. So one of the key, I broke these up into kind of composition and process related. So one of the key ones really is for the enteric polymers anyway. As Chris mentioned, at least in the lower pH, in fed or fasted gastric pHs, these the polymers are relatively inert, and what that means is they don't gel, and that means that the excipients that we use can really have their full impact, the fillers and the disintegrants, to allow us to give this fast disintegration. So our goal really is to get a fast macro tablet disintegration, such that also the other part B of that is that the particle size that these tablets disintegrate into are as small as possible. So it helps to be using these somewhat inert, non-gelling polymers for that. Again, we use very conventional fillers, disintegrants, lubricants. We can get high loading. We very commonly are 50%. I would say up to 70% is within our working range. 70% and below, and 50% below is very straightforward. On the process side, the particles are spherical, I mean, relative to crystalline API, which of course, the space of all API is all over the board on morphology and size. The size here is always going to be relatively spherical, relatively narrow particle size distribution. They're not aggregated. The particle size from from the very first early batches up through commercial encompass a range of maybe 20 to 100 microns, most common. The dry granulation, they said, is possible, but if we can, if the loading of the dose and the properties are such that we can do a direct compression, that is our preferred process. One important thing is the properties of the particles. Compaction properties they are actually very good. They're I would say they're similar to excipients, maybe somewhere between Avicel and lactose on compressibility, ductility, and bond. So that rarely is a problem, compressibility. Until we get to, if we had a combination of extremely high API and extremely high loading in the tablet, then that's something we will look at. But generally, it's not a major concern. We don't see much sensitivity to process conditions. Again, this is a little bit limited to the enterics. That can change. The native properties of the API are masked, as Chris mentioned. Some of the practical aspects of that is one of the issues that we really never deal with is API sticking on the tablet tooling. The hygroscopicity is low, so we don't have to have any specific conditions for processing and handling of the powders. As I mentioned earlier, film coating is possible. On this slide here, I've shown what would be sort of a black box formulation that works for an awful lot of SDDs. There are some subtleties into this, but if you just look at the composition and the amounts, you'll see again they're quite similar to what you do for conventional API. I'll just kind of go through the comments here. So the ranges in here in the tablet, I mentioned the ranges are maybe 70% and below, and certainly 50% is common. We usually use a mixture of a ductile and a brittle filler. There are some exceptions to that, but it is the most common here. One thing we find, and generally we'll get better performance by using the smaller grades of these. Again, this is trying to maximize the surface area of the excipients relative to the spray-dried dispersion. We're trying to minimize the spray-dried dispersion particle interfaces within the tablet. So small particle size fillers generally is better, especially if we're dry granulating. That generally is not a big issue. Disintegrants are normally at higher level than what's used in conventional IR. Oftentimes we'll add a glidant. Sometimes intragranular as well. There's some functional reasons at times to do that, but just as a flow aid as well, and increasing the density, bump density, and any of the standard lubricants and coatings. As Chris mentioned, getting into customizing for particular APIs, different fillers on some occasions. Sometimes we'll have different additives. If we really have log P, we might have some surfactants. So there are some variations to this, but generally speaking, this would be a starting point for most SDD tablet formulations. The next slides I'm going to talk about are our process and formulation development. I've got some flow charts here that we utilize. We normally are limited in the API amount that we have available. So I mentioned earlier, we do break up the selection of the tablet composition from the process development somewhat cleanly. So this first slide here is kind of a screening mode. These would be small batches that we make at bench scale. We don't use actual automated processing equipment for most of this. We do, again, what this is developed over time as what we can do for screening. And the primary screening metric that we use here, given the dose that we'll have and the tablet size constraints, we're selecting the excipients and the levels, and whether or not it's a direct compression or a dry granulation at this stage. And primarily it's disintegration that we use early, and then dissolution would be the confirmation. That's the main thing we're doing here at this point. So we're not involving the process parameters a whole lot. So we will generally slug on a single layer tablet press. We will mill these manually, either with a mortar and pestle, or sometimes use a small co-mill cone screen. At this point, we're not, again, I mentioned it's not very critical on particle size. We found the granule particle size is not critical to dissolution, so we don't do a whole lot in the screening mode here. We'll blend with extra granular in some cases, and then compress tablets, usually on a single layer press at this stage. So again, this is very minimal, 10, maybe 5 to 30 gram batch sizes for these tablets. And we'll be looking at disintegration and dissolution as the metric here in the dissolution test that Chris showed. And I'm going to spend a little bit of time in the next 2 slides talking about the disintegration because, okay. So we could just do a visual. Actually, I think I'm going to skip to this slide here first. We can do a visual assessment in just a standard disintegration apparatus. And we're looking for small particle size, disintegration fast into a small particle size. Actually, Chris coined these 2 terms that we use around here. Routinely, we call it a blizzard. If we have in the dissolution flask, when we do this disintegration, if they disintegrate quickly into very small particles, it ends up being very fine particles, and it kind of looks like a blizzard. The converse to that is a snow globe. In this case, we get disintegration, still might be rapid, but the particle size is not that small, and there's still some aggregates there, and sometimes it can look like a snow globe that you'll invert with these aggregates. So we're trying to have these small particle size. So that's a qualitative screen. A little more quantitative version of that is we do use an FBRM to look at this, and on the right-hand side would show that some data. Let me go back a slide here. FBRM. I wanted to acknowledge Kerry and David Sperry at Eli Lilly. Old friend and colleague David and Kerry helped us out on getting this set up, and Justin Huey was here before he went on from Texas to Bival to develop this method. So we do use an FBRM. Of course, FBRM measures what it gives us out of this is particle size versus time. And this shows our installation in a dissolution apparatus. We typically are not simultaneously measuring dissolution. This is just disintegration. And on this slide on the upper right-hand corner, what you'll see would be when we do compare the dissolution, the large particle size would give us lower dissolution, and that would be manifested by larger particles. On the middle screen there, the green curve would be larger particles. And then on the bottom, actually the number of counts is also lower for the slower formulation. So once we've selected a formulation, then we move into the process development. So once we've identified a lead composition, we still stay at this small scale for the very early prototyping. And here, though, we're looking at the compressibility and identifying the solid fraction and the particle size that might be targets for when we scale to the roller compaction. And if you just look down the bottom left there, we will look at the compression profile. Here we'll be on the lower end of our pre-granulation blend. We'll generally target a tensile strength of our slugs of 0.5 solid fraction for the slugs, and then we will mill these into this 100 to 300 micron range, and then again do the hardness compression profile, which will be shown on the bottom right-hand side there. So what we're assuring at this point is that we have adequate compressibility of our dry granulation, and we will target at this point is to assure that our final blend has a compressibility of about 2 megapascals. So we'd like to be able to achieve up to 3 megapascals to give us some flexibility here. So that would be our target at this small scale. Maybe 1 to 3 megapascals of our final blend is what we're targeting here. If we don't achieve that, then we would go back to alter the composition or the dry granulation solid fraction or the density, so that we would so that more compressibility remains in the total formulation. So again, this is all at small scale at 5 to 30 grams. So at this point, we've identified, and of course, if this is a direct compression, we're just doing a standard compression profile, which would be on the bottom right-hand curve here. And as I mentioned, we very rarely have issues with this, but this is just we still generate this because then it gives us our guidance for the roller compaction settings to achieve the desired ribbon. Of course, what we found is that we actually have a very good agreement of being able to scale from these slugs to ribbons using the solid fraction technique. And then, so what we leave really at this stage is the milling development once we move on to the next step, which is shown on here. So now we've gone from a bench scale onto real processing equipment, a roller compactor, and mills and tablet presses. So again, a little bit of confirmation here on the left-hand side. We would do a blend. Actually, that shouldn't be a turbulator. We should probably have a cone mill at this stage or a PK blender. We would normally be above 400 grams here to get on a roller compactor. And that has to do more with the milling than forming the ribbons as a minimum batch size. But again, so now we have these compression profiles from previously. What we'll do at this point is also generate, because it's a little difficult to measure tensile strength of ribbons. So what we'll do is develop a calibration curve of the tensile strength of our slugs versus the solid fraction, which is an easier thing to measure. We use a geopic. We can also do this by physical measurements when we're using flat face round tooling. So what we end up with is a calibration curve of tensile strength versus solid fraction. Solid fraction then becomes something that we can measure easily, again using an envelope densitometer like this geopic, and the AccuPic here is for the helium pycnometer, which is another thing that's needed to calculate the actual solid fraction. So then we move on. So we have the parameters from our compression profile to determine the roll forces to achieve our ribbons of the desired density or solid fraction. So we'll run those at startup to make sure that we achieve our target solid fraction that we identified at our bench scale. And what we're really left then with is identifying the milling parameters to give us the desired particle size of the milled granules. So that is a little less predictive, so we end up doing anywhere from 1 to 5 conditions typically to achieve a range of granule sizes that we then will evaluate for tableting in some cases, or we'll just do, and more often than not, we'll take those at small scale to assess whether there's an impact at on our dissolution performance and compressibility. So out of all of this, we now have the roller compaction, roll forces, and the feed speeds and the ribbon thicknesses identified for what we need. We've identified the mill parameters to give us the target particle size of the granules. Of course, we characterize these for density and flow and morphology and so forth. But that then allows us to move into the tableting. The tableting process development is just straightforward. Oh, sorry, need to advance the slides. Thank you. There's really not a whole lot to talk about here. The tooling, I guess I'll just point out again that we don't need to use coated tooling. Rarely, if ever, do we have sticking issues because the polymer properties mask the inherent nature of some of these hydrophobic drugs that tend to stick. So at this point, we really just are looking at the specifics of the tableting process. So we're showing here our Course XM12 presses. At this point, we're really looking. Oftentimes, we're balancing the minimum batch size and the feed frames and the residence times more. So it's more of the typical things here, nothing specific to the SDD that we are optimizing. But at this point, we're not expecting surprises on dissolution and disintegration because we've really done this at the small scale. But obviously, we do the confirmation, and then we're looking at things like content uniformity and assay become the metrics that we're looking at during the tablet development here. If we do see a surprise at this point in our dissolution or disintegration, more often than not, we'll end up being able to identify that as something to do with a long residence time in a feed frame, a lubrication, or something like that that might require a final tweak. Again, our primary metric here for performance is this two-stage dissolution that's shown under the dissolution graph there, the flask. So, and we generally are always comparing this to a suspension, as Chris mentioned. And the reason we do that is actually it's while that sounds like kind of a lofty goal, that is our goal, is to achieve the performance of an immediate release tablet that's similar to a suspension of the native SDD particles. So on this graph, I'm showing some data that we can present publicly here. This is a compound that had an aqueous solubility of 60 micrograms per mil. It was formulated into an SDD at 25% loading and 50% loading into an HPMCAS polymer. The drug was non-ionizable, a log P of 2, a melting point of 238. Compared to a crystalline drug into a crystalline API, 10 microns, the human PK is shown here for immediate release. You can see the solid dispersion gave more than 5 times the AUC. So that also is shown the time course is shown on the bottom left-hand side here in human volunteers, and then on the right-hand side are 2 tablets, one with the 25% active and the 50% active solid dispersion. And you can see that those compare quite favorably as well. So this is obviously a case of a success, but I can say that when we're in this platform of using the technologies with the enteric polymers in particular, we can match the suspensions. When we are using non-enteric polymers, sometimes it'll be a little bit slower, but achieving the AUC, I would say, is the expectation there. The Cmax and Tmax might be slightly suppressed depending on the overall on the final formulation. So that was mostly what I wanted to say on immediate release. I'm going to just a couple very high-level slides on modified release. So it might be implied here. We really do treat the solid as as a tablet formulator we kind of are able to treat the spray-dried dispersion as almost the salt form of a drug, of an API. It's just a modified API that, that's diluted. So, dose becomes probably the one thing that we tend to, spend most of our time optimize, because there's a payload of polymer that goes along with that. But the two controlled release technologies that this technology that we've explored and works really quite well is osmotic tablet technology. We happen to like that because of its robustness, and it's again, it's platform technology. And I have time here, I'll try to go through a case study that's showing how we do this. But we really, in this case here, this is similar to the ALSA push-pull technology. There's some subtle differences in the technology, but it works similarly. Bilayer tablet that has a swelling layer and then a drug layer. In this case, the white or upper layer contains the drug. In this case, it's containing the SDD. Generally, about 30% of that of that drug layer is the SDD, as a rough rule, or less. So we just apply the standard technology, the laser drill, and what you see on the right-hand side is this behaves as expected for this technology, where the drug hydrates and the drug is entrained, and it's extruded out, laser drilled orifice. And the bottom right here shows the range of dissolution profiles for that are obtained from typically this osmotic will give zero order profiles like this, and we can control the release rate over a pretty wide range. Also, we can take these spray-dried okay, we had the animation on there, I didn't realize. So here we're just showing putting the spray-dried, well, this is the in vivo results from that. On the left-hand side here is the in vitro top. On the right would be the in vivo results, and of course we're seeing the modified release. And as typical with an osmotic system, there is a lag period that's associated with the activation of the dosage form. In this particular case, we were able to achieve the fed-fasted effect was mitigated by using this controlled release technology. We can also take the spray-dried polymers and incorporate those into a standard hydrophilic gel matrix tablet, commonly HPMC, but also can put these in PEO or an HPC or other hydrophilic matrix. Probably the key thing to note about this, as opposed to a crystalline drug, is now the drug or the dispersion itself is a polymer that has some viscosity, and there tends to be some interaction of this polymer with these hydrophilic matrix, and it tends to form a fairly robust gel. So what we end up with is the equivalent of a gel matrix tablet that has a poorly soluble compound. Of course, the drug is poorly soluble, but even using these polymers does not render it a different mechanism. So it does maintain this erosion mechanism. What we tend to see is they tend to be a little bit more robust films than just the regular matrix because of this interaction. And again, as with we can modulate with different molecular weights and loadings. The in vitro and the in vivo correlation showing on the bottom and left here. That's a human study with an IR and two different CR dosage forms using the matrix technology. How much time do I have? 1 minute. Okay, that's not very long. I guess I was gonna go through. I have some backup slides that, if you request them, I guess it will show a case study that shows kind of putting this together on using the small-scale benchtop approach, which we also use for the osmotic to develop the drug layer, but then tagging on the bilayer component of that and the osmotic coating. I think it's all similar and then showing the kind of some of the metrics of a final tablet made in clinical manufacturing. So anyway, I'll leave that for you to peruse at your own leisure later if you're interested. So the conclusions here, just very generically, obviously Chris has pointed out, and I think it's pretty well established, this general technology works very well for enabling poorly soluble compounds. The main message I want to leave here is that the properties of the SDD particles themselves are really highly amenable to incorporating into immediate and modified release tablets using common excipients and processing technologies. They are, and the dry granulation and the direct compression, are intrinsically continuous unit operations that are very scalable, which is very nice. Just because of the platform nature of the polymer properties, we can count on the properties and the API properties, or I should say the drug product intermediate properties, remaining the same throughout the life cycle of the development and commercial. It allows us to have these material sparing approaches that we can count on for developing robust formulations with minimal amounts of material. When properly formulated, we get performance from immediate release tablets that's similar to dosing of suspensions of the drug product intermediate. And the loading is high. Loading defined here as loading of the drug product intermediate into the tablet. I would say, for a 50% loaded SDD, 250 milligrams in a dosage unit is what is our typical target here. If it's down to 25, we're probably our target might be around 150 milligrams. If we're fortunate enough that the properties are such that we can go above, we can approach up to 400 milligrams in a single tablet in some cases. I think that's all I had. This is like our ending slide. I guess I'll turn this over for any questions that may have come through here in the time that we're waiting, or that time that Chris and I have been talking. Do we have any, Phoenix, that, or the moderator is going to, I guess the moderator has any questions that have been coming in.
That's right, yeah.
So she may be, she'll ask the questions.
For your insightful presentations. We did receive quite a number of questions, so we'll now move on into the Q&A part of our program. At this point, I would like to invite our audience to continue sending their questions or comments that they would like to address to the speakers through the question function located in the question panel on the right-hand side of your screen. I've already received a few questions, so I'll start with those. The first question is, how do you determine whether to do direct compression or dry granulation? Chris, Randy, if you would like to address that.
Yeah, so I'll answer that from two levels. The first one from a performance standpoint, where I'm defining performance here like the dissolution performance. From that standpoint, we view there as very little difference. Generally speaking, they're equivalent. There are some cases where dry granulation will afford us some other flexibility that direct compression won't. That isn't so true for the enteric polymers. For the non-enteric polymers, we will manipulate the ratio of intragranular and extragranular to, in some cases, so for those, more often than not, we will lean towards using dry granulation. But for the enteric polymers, in particular HPMCAS, we would consider them interchangeable. So that's the performance side of it. From the processing side, it really gets down to the dose and the properties of the SDD. So if the lower the dose, the higher the chances of direct compression, because it really gets down to flow. The SDD particles themselves flow okay, but not great as a general rule. They're not as good as fast flow lactose. They're not as bad as as pore-flowing APIs. There's somewhere in between, depending on the loading and the solvent system. That is something that we optimize as we progress. So the density and the size of the SDD particle and the dose are the three things on the process side that determine whether we go direct compression or not. Yep.
Thank you. Our next question is, what inert non-gelling polymers are used in the tablets?
That's a good question there. I think that has to do there. Typically, we do not, for immediate release materials, include additional polymers into the tablets there. I think that question is probably partially related to the discussion there where we've talked about the dispersions, having a potential to gel in the capsule, and then, one of the natures that Randy mentioned is that they were inert. And I think what should have probably a little more clarified there is that on early time scales that the dispersions take a little bit of time before they're going to, in the gastric material, before they would wet and begin to soften and have a potential to gel up there. And so what we do is we take advantage of the time in the gastric there, and to use the tablets to rapidly push the SDDs apart such that you can reform the suspension in the stomach. Whereas in a capsule and formulated, the capsule itself holds the material tight while it's wetting, and then allows it to, over those few minutes, to gel up and then create a larger particle that takes a long time to gel, or to dissolve. Excuse me. I hope I answered that question well.
Yes, thank you. Our next question is, if using SDD formulated granules in the capsule for immediate release in early phase?
Yeah. The last part of that question looks like, is there a preference for HPMC versus gelatin capsules?
Yes.
Okay. So we'll make the assumption here that they're formulated, putting granules in, and they disperse rapidly. So generally speaking, HPMC capsules do dissolve just a little bit slower than gelatin. That's at least been our experience. So there can be situations, depending on the granule formulation, where a gelatin might dissolve faster, therefore the contents be able to disperse and more quickly and work a little bit better. But as a general rule, I'd say there's not a substantial difference between the two. One other consideration is HPMC in of itself, in some cases, can be a crystallization inhibitor. So in that, there might be cases, select cases, where HPMC may enhance the supersaturation sustainment.
Right, thank you. The fourth question is, which vehicle are you preparing suspension formulations of SDD?
So when we're preparing suspensions, they're typically aqueous suspensions at high concentration, and just typically in a methicel solution is what we use.
Yeah, I mean, one thing to point out, there is a little bit of a technique there that
in preparing
it, just a caution here that in preparing that suspension, the same thing that can happen with capsules if you don't prepare the suspension right, is you can cause aggregation and gelling of the SDD particles.
So to point out, if you were just to take the powder and directly add it into the liquid, you could have a problem there with getting the material dispersed. And so there is a technique for the preparation, but the suspension that we ultimately make up is just a simple aqueous methicel.
Yeah, so it's basically a wetting procedure, pre-wetting procedure, and then diluting. So.
Right. Moving on to our next question, it says, do you find any incompatibility of polyox with HPMCAS do you find that due to amorphous state of API in SDD, it could become more reactive and may affect polyox in drug layer of osmotic swallowable core tablet?
Polyox interacting with the, I don't know if we've seen any interaction with polyox. You do have to worry sometimes about the potentials of some of the peroxides that can be left over in the PEO with chemical stability of your API, if that's a potential bit there. But in terms of the PEO interacting with the HPMCAS in an osmotic tablet, no, I don't think we've run into any issues with that.
Yeah, another way to interpret the question was, you know, by it, the drug, of course, is amorphous, and the which, you know, intrinsically then is going to be more reactive than if it were crystalline in the SDD. So generally speaking, though, that, you know, I'm not sure we have a case where just by being amorphous has been the difference in a PEO formulation. It would either just be substantial chemical degradation due to the PEO. Of course, we do an excipient compatibility up front to it, whether we determine this technology is active or not. And to do that, oftentimes we will just use crystalline drugs for that screening, although, you know, we don't, just because we don't see the amorphous being substantially more reactive.
Right, thank you. Our next question is, are there any evidences that compression forces break the matrix structure of a solid dispersion of amorphous substance?
Yeah, that's a good question. Our experience has been with HPMCAS dispersions that when they go through the tableting process that we do not see form change related to the amorphous material there. And I guess the question is also in terms of long-term stability, you know, is there any differences there? I can sort of add to that. And that if the dispersion is stable as a powder, it has been, in our experience, stable as a tablet formulation. I believe that there are some cases where dispersions can become unstable with compression, but those are typically with other dispersion polymer.
Right. Thank you so much for answering those questions. I'm sure our attendees have found this presentation quite informative. We have come to the end of our presentation. I thank everyone for participating in today's conference. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event. Please join us in thanking our speakers, Chris Craig and Randy Wald from Bend Research. Thank you so much, Chris. Thank you, Randy. We hope you found this conference informative. A survey window will pop up on your screens. Your participation is appreciated as it will help us in developing our future webinars. You'll also receive an email with an access to the recording for today's session. If you would like to share it with your colleagues, please reply to the email and let us know. And for any, if you want the presentation to be forwarded to your colleagues, please let us know by email and we'll have it forwarded to them. Thanks again for joining us, and we hope you have a great day. Thank you, and bye-bye.